Okay, enzymes and energy, part two. So we need to talk about activation energy barriers. So every chemical reaction between molecules, so remember what we said a chemical reaction was, it was going to be making or breaking bonds. So every chemical reaction is that whole, we're all focused around the electrons. The electrons are what's going on and moving around, and that's what's happening here. So when I make or break a bond, that's how a chemical reaction occurs. And they all need some sort of initial energy to start that chemical reaction. Okay? That initial energy is called the free energy of activation or activation energy. I'll always refer to it as activation energy. I'll write it E sub capital A, just like it's here on the slide. And oftentimes activation energy comes in the form of heat from the surroundings. So that's important to know. All right. So enzymes catalyze or speed up reactions by lowering how much activation energy is needed. Let me say that again. Enzymes make a reaction go faster. They catalyze a reaction by allowing the activation energy to be lower, to need less initial energy to drive this. I need less heat to make this happen. Okay? This next thing that I'm about to say is huge. Huge, 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 huge. Highlight it, draw stars around it, circle it, big purple arrows. Whatever you do, do it. But this is important. Enzymes do not affect the change in free energy. I don't alter the initial energy or the free energy contained within the products, and I don't alter the free energy contained within the reactants. Those things do not change. The only thing that changes when we're talking about an enzyme is the amount of energy needed to move from reactant to product. I'm using less when an enzyme is present. It looks like this. So that black line goes, I'm going from reactants here to products here, yes? This is my free energy, just like we did before. This is progress of the reaction. My reactants have this amount of free energy when they start. My products have this amount of free energy at the end. The difference between them is right here. The difference between the reactant energy and the product energy is right here. Okay? Now, if I don't have an enzyme present, this reaction still occurs. It just takes longer. And it takes longer because I need more energy. I need more energy to drive this. I need more activation energy to come up here before I can push over and generate products. That's my activation energy that I need without an enzyme. If I have an enzyme present, I'm going to talk about this red line. See this red line? I come to the peak much more quickly. I come to it at at a lower free energy level. I needed this much without the enzyme. I need this much with the enzyme. And so I come to it at a lower energy level, and then I proceed to products. So the difference here, the thing that changed, was the amount of activation energy without an enzyme to the amount of activation with an enzyme. It's less. Look at this. This is really, 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 really key. Right here, my reactants had the same amount of free energy with or without the presence of the enzyme. It didn't change the reactant. Nothing changed about it. It's still the reactant. Over here, my products didn't change. They don't have more or less free energy. They are what they are. It's just that I was able to get from reactant to product more quickly because I didn't have to put as much energy into the equation. Okay, I've got some questions for you. I'll count to 10. You pause, do what you got to do, and then we'll answer. How'd you do? Another set. Oh, I was wrong. I fibbed. It comes later. All right. So we have to talk about enzymes. Okay, we know what they do. We know that they're proteins, so we've got this vague idea, okay, I've got this specialized protein, great, and it's going to lower this thing that I don't really know what it is, but Dr. P says it's the activation energy, and I've memorized that that means how much energy I need for the reaction to go, and okay, super duper, that's great, but, okay. So we're talking about a chemical reaction. We're going to go from reactants to products, right? That's a chemical reaction. I've already taught you that. You know that. Okay, so in this case, whenever there's an enzyme present, because science has to name things differently, we're going to change the name of reactant and we're going to call it a substrate. It's the same thing it was before when we called it a reactant. It's the exact same thing. It's just that now, because we have an enzyme there helping us out, we can't call it a reactant anymore. We have to call it a substrate. Okay? And so what happens is the enzyme binds to the substrate, and it forms this complex. It forms this complex molecule. And it doesn't just bind in any old place. The substrate binds at a specific space. It's a lock and key sort of thing. It can only fit into this one spot. And this one special spot is called the active site. So the active site is this region on the enzyme that the substrate binds to. Okay? And this whole lock and key thing is the concept of an induced fit. Okay? So the induced fit of a substrate, so when that substrate binds to that enzyme at that specific active site, right, it enhances that enzyme's ability to catalyze the reaction. If the substrate doesn't bind to the enzyme just hangs out. It's not doing anything. It's just this protein floating around. But if it binds to the substrate, the correct substrate, then I have something called induced fit. And then I can see the process of catalysis. So the substrate is the little pink thing, the big blue protein thing is the enzyme, and when that substrate binds at the active site to the enzyme undergoes a change. It changes its bonds temporarily, and we get that image on the right. Okay, so remember I said all along I'm going to lower the activation energy, right? I need less energy to make this happen. Well, I can do that through one of 4 ways. Okay, so I can orient the substrates correctly. I can line up those pieces to make the product. It's like having the pieces to a puzzle but not having them lined up the right way, and the enzyme comes in and lines them up the right way, and boom, they're oriented correctly and they just slide right into that puzzle. Okay, that's one thing an enzyme can do. The other thing an enzyme can do is by forming those bonds between the substrate and the enzyme, I can put strain on other bonds within the substrate and allow those bonds to break more easily, and then we can reform into the correct products more easily. Third thing we can do is we can provide a favorable microenvironment. So for instance, let's say that I need a specific or a particular pH. The presence of that enzyme and the amino acids within that enzyme can give us an environment, a microenvironment, an environment at that active site that is perfect for this reaction to carry out. And then the fourth item is covalently bonding to the substrate. So whenever I take over and move things, break molecules away and covalently bond to the substrate, I'm going to change its structure, right? I can't just break a bond and bind to something without changing other stuff. Remember what we said, structure follows function. So if I change those bonds, I'm going to change the structure, and thereby I'm going to get my products more easily. Here's the visual for those in my group who love the visual. It's numbered out. It's got cute little terms that go with it. If it's confusing at all, ask me in class, hit me up on email, give me a call, whatever, and we'll talk it through. Okay, there's your whiteboard I promised you. More true or falses. I'll count to 10, you'll do your work, we'll move on. How'd you do? Okay, we've got like, I don't know, 8, 10 slides left, I don't know, something like that, maybe 7. Okay, so enzyme activity on local conditions. So this is kind of what we said. So enzymes can be affected though. So I'm not talking about what the enzymes do, I'm talking about what affects the enzymes. what makes an enzyme work or not work. So environmental factors. We talked about this way back in macromolecules when we talked about denaturing proteins. So because an enzyme is a protein, right? All enzymes are proteins, not all proteins are enzymes. Because an enzyme is a protein, if I denature that protein, if I denature that enzyme with like temperature or pH, right? Then I'm going to change the structure of that enzyme and I'm going to change its ability. Typically, if I denature an enzymatic protein, it doesn't do its job anymore. It changes its structure and it can't. It doesn't have the right substrate binding site or the active site. It can't bind the substrate. It doesn't do its job. And there are certain chemicals that specifically influence that enzyme, good or bad. So we can have enzymes who are positively influenced by the presence of a chemical, and then they work even better, or we can have them impacted negatively, where the presence of this chemical prevents the enzyme from doing its work. Here's what we just said about temperature and pH. Every enzyme has its optimal pH or its optimal temperature and pH than which it can function. That doesn't have to be neutral. That doesn't have to be body temp. Certain enzymes work better at higher or lower temperatures. Other enzymes work better at higher or lower pH values. The enzymes in the stomach, in the gastric juice, function very well at an acidic environment, right? But you stick that enzyme in the small intestine or even the large intestine where you're in a basic environment, and that enzyme is no longer going to be able to work. So key to understanding enzymes is knowing that they have optimals. They have situations in which they function their best. Just the way you and I have situations in which we function at our best, so do enzymes. Cofactors. Cofactors are non-proteins. They're enzyme helpers. They're not an enzyme, they're just a helper. They're a booster, okay? Cofactors can be organic or they can be inorganic. So we can have a metal in ionic form, and that can be a cofactor. If the cofactor, however, happens to be organic, we call it a coenzyme. The most common example of coenzymes are vitamins. And what cofactors do is they help enzymes do their job better, okay? Opposite of that are enzyme inhibitors. Okay, so coenzymes are enzyme promoters. These guys are going to be enzyme inhibitors, and I can have a competitive inhibitor and I can have a non-competitive inhibitor. So remember that substrate binds to the active site, right? So the active site is on the enzyme, the substrate comes along, binds to it, and bada boom, bada bing, catalysis happens and we get a quicker reaction. If somebody else comes in and hogs up that binding site, then the substrate can't bind there. And if the substrate can't bind because somebody else is sitting in its seat, then the enzyme doesn't work. The enzyme is inhibited. Okay? So that's a competitive inhibitor. A competitive inhibitor binds to the active site and prevents the substrate from binding there, and thereby prevents the enzyme from doing its job. And we have an image of that on the next slide. A non-competitive inhibitor binds to some other part of the enzyme. It does not bind at the active site. It binds somewhere else. But because it chemically bonds somewhere else, it rearranges the structure. And by changing all of those bonds in that protein, it causes the active site to change its shape. The bonds get rearranged and now it's not quite the same. Okay, and now what happens is the substrate can't bind to it because it's not the same shape. Okay, like I said, I'll show you in the next picture. But some examples of inhibitors are things like toxins, poisons, pesticides, antibiotics. That's exactly how antibiotics work against bacteria. They prevent them from reproducing. They break down in some cases, they break down their cell walls. They prevent them from undergoing brain stopped. They prevent them from reproducing, any of that sort of thing. So here's your image that I promised you. So on the left, normal binding. Substrate is teal, enzyme is purple. Bada boom, bada bing, they hook up. Isn't that awesome? Catalysis happens. B is competitive inhibition, and that blue guy is a competitive inhibitor. He chunks in and sits down where that substrate needs to go, and now that substrate can't bind there. The lock and key doesn't work. So that's a competitive inhibition. And a non-competitive inhibition is at letter C, where on the opposite side of the enzyme, and it doesn't have to be the opposite, it could be right next to it, it's just in this image that it's opposite. So at some other site on the enzyme, somebody else comes in and binds, causes a rearrange of bonds and structure within that protein, and now the substrate doesn't fit the way it's supposed to into the active site. If we didn't have the ability to turn on and off enzymes, we would have chemical stinking chaos. Metabolic pathways would be shooting off right and left. We'd have no way to stop them. Everybody would be running at max speed all the time, no matter what. And that's not going to work. That's just not going to work. So we have to regulate those metabolic pathways, okay? And a cell does this by turning on and off the genes that encode for specific enzymes. So by regulating the enzymes, we regulate the chemical reactions within the cell. And how do we do that? We have structures to help bring order. We have some enzymes that act as structural components. So enzymes, what do enzymes do overall? They can help with the metabolic pathways. They can determine which pathways are going at what time. How do they do that? Well, based on how much enzyme is produced from the DNA. They can act as structural proteins. Remember, we had enzymes that were embedded into the cell membranes, into the plasma membranes. In eukaryotic cells, so any of those cells with a membrane-bound nucleus, organelles, those guys, some of those enzymes will reside specifically in specific organelles. For example, enzymes for cellular respiration, the enzymes that drive the process to make ATP, a lot of those live in the mitochondria. They're not just anywhere in the cell, they're hanging out in the mitochondria where we do this work. So those are some of the things. Our next chapter, I don't know what our next chapter is. I don't remember what our next chapter is. But that's it for enzymes and energy, or energy and enzymes. That's your part 2. And I'll get these loaded up for you. All right, later.